Hi everyone, my name is Björn Pokorny, and my presentation today is about the molecular techniques used in a study to identify the role of bacteriophages in atopic dermatitis. The title of my article is The Phagiome in Normal and Inflamed Human Skin, and it was published this year in the Science Advances journal. So first, a quick overview of what atopic dermatitis actually is. It can be described as a chronic inflammatory skin condition that is not contagious, and it is actually the most common type of eczema. It affects about 10 to 30% of children and 2 to 10% of adults. The causes can be genetic, immunological, and environmental. And it's basically caused by a defective skin barrier that becomes extremely susceptible to dryness and environmental factors. The main symptoms are inflammation, rashes, thickening and hardening of the skin, and itching, which can lead to bleeding of the skin. Of course, this leads to an increased risk of infection. It can be managed with daily skincare, anti-inflammatory therapies, and avoiding triggers. And this will be depending on the severity of the person and what their personal triggers are, because it will be different for everybody. The main focus of the study was to implicate which role the bacteriophages play when it comes to atopic dermatitis. They can be described as viruses that infect and replicate inside bacteria, and they affect the composition and evolution of the bacterial communities. They can contain either DNA or RNA, which can be either double or single-stranded, and genomically and morphologically they are very diverse. As you can see in the image, there is a schematic and then also a microscopic view. And this is just the one that they've encountered most often, but they can look very different from this as well. The first technique they used in the study was shotgun metagenomic sequencing. It's a type of next-generation sequencing, which means that it is high throughput and they can sequence many fragments simultaneously. It is fast and more cost-efficient. Shotgun metagenomic sequencing aims to capture or sequence the complete genome opposed to just one fragment. The DNA is fragmented into random pieces, so it's not specific. And all these fragments are read, and then a short sequence of nucleotide is generated. So there will be overlapping sequences, and by matching these overlapping sequences, it is able to assemble it into longer, continuous sequences, which are referred to as contigs throughout the paper. Eventually, they were able to obtain a viral metagenome, which was basically reconstructed from all the genomic data, which means basically that all the DNA that was in the sample, the human DNA, the bacterial DNA, and the viral DNA, from that they were able to obtain or analyze or pick out just the viral DNA. The other technique used in the study was real-time quantitative polymerase chain reaction, or RT-qPCR, which basically amplifies a specific DNA or RNA target sequence, which allows for the calculation of the initial concentration in the sample. And it also offers real-time monitoring for continuous quantification. On the image, you can see the one-step procedure, which is the RT-qPCR, and it also shows the two-step sequence, which is just regular PCR. So the RT-qPCR is faster and more efficient because it's just one step. So this was a case control study with cases, meaning patients that were affected with atopic dermatitis, and a healthy control group who were not affected. To try to adjust for the confounding factors, they excluded several types of participants, and all the samples were collected between 2020 and 2022. Each participant had 2 skin swabs taken at a specific site from a specific area to try to do the study as consistent as possible. So the results showed that there was a strong negative correlation between certain types of bacteria, in this case P. agnes and S. aureus, in healthy and affected cases. And it also shows that the patients containing S. aureus had a higher extent of inflammation. So in image B, you see the control versus the affected group. And in image C, you can see which type of bacteria caused the most inflammation, from red to blue, which is the least type of inflammation. So on the previous slide, we saw which bacteria was more likely to cause inflammation. And on this slide, we are going to look at the actual bacteriophages. So they were able to find 164 viral metagenomes, including 11 complete ones. The most significant one was, maybe expectedly, the phage that was found in the Staphylococcus aureus bacteria. It was named its VMAC14. And people carrying this prophage had an increased inflammation or severity in their atopic dermatitis. In VMAC4, which was a phage found in S. epidermidis, the phage numbers decreased with increased inflammation in atopic dermatitis, while the actual bacteria, S. epidermidis, increased in abundance. VMAC8 was also found to increase with inflammatory severity, but the host mapping was incomplete, so they're not quite sure which host it comes from. The results did not correlate completely. So on image A and B, you can see the severity with in combination with image B. You can see which VMAC or which phage metagenome corresponds to the severity of inflammation. And in the table next to it, you can see the regression analysis that they did, which basically shows how significant their results are. And the first three, VMAC4, VMAC14, and VMAC8, those are the three VMACs that were mainly found to have significant implications, and also had high quality hits that could be confirmed and most likely play a role in atopic dermatitis inflammation. So in conclusion, this was a first of its kind study, because they did not use any culturing. They only used molecular techniques. There is still very little known about the skin phageome, but the results encourage further research and also the further development of different techniques to investigate more. From these results, we saw that some phages may have a positive effect on actually healthy skin, but some may also benefit the bacteria in ways to increase the severity of atopic dermatitis. If anything, it also implicates that there is room for phage therapy in the future aimed at relieving or maybe even curing atopic dermatitis by using or leveraging the natural role and functions of these phages. These are my references that I used. I hope you enjoyed the presentation, or at least found it informative. Thank you for listening.